Thank you to the Built with Biology Conference for hosting us today, and Frank Tate for the invitation. It's wonderful to speak with you all at such an inflection point within biotech as we emerge from a post-pandemic world and truly embark on what is shaping up to be the century of biology. As we're on an eve of a revolution, I think in life sciences, it's amazing to see the further integration of technology and biology come together, and treatments truly get hyper-specialized in this, and finally the aspects of medicine catch up to our unique aspects of biology. And I think today, as we highlight that with the precision medicine, hopefully give a little more peek of behind the scenes here. I'm Pulido, founder and general partner at Alix Ventures, an early stage VC firm here based in San Francisco, focused on bioengineering companies enabling advances in human health, and founder of BIOS, a life sciences content community platform, and joined today by two of our portfolio companies and their amazing leaders, Jason Springs, co-founder and CEO of Endpoint Health, and Gary Yeung, CFO and president of BioPharma at Xilis. Gary, if you could kick things off for us and give a brief introduction of yourself and your work at Xilis.
Sure. So Gary Yeung, as Chas mentioned to you, currently I'm with Xilis. Prior to that, I have been in the industry for 25 years. 14 years with Genentech, done everything in a larger biopharma company could do, research, development, and commercializations. In the last 7 years, I spent most of my time building a pretty successful biotech company, including 3 that went IPO. One is Guardant Health, it's about $7 billion market cap. Another one is Inixon Biosciences, the neuroscience space here. And the last one before Xilis is called Erasca, stands for Erase Cancer. Employee number 2, in 3 years, we raised $300 million, went IPO. So with the IPO money now, I do have some financial freedom. So something that you guys could aspire to, okay? So next.
Thanks, Gary, for joining us. And Jason, if you can share a bit about your background and your journey with Endpoint Health.
Sure, thanks. So Jason Springs, co-founder, CEO, and board member at Endpoint Health. So spent about 20 years not all in the healthcare industry, always in complex regulated environments. Started in aerospace, moved to diagnostics, now in pharma, actually kind of integrating some diagnostic products there. Always enjoyed looking at really complex problems where regulators are involved. Fun problems to solve, really important when you do. Previously started a company called GeneWeave, so that was a Cornell University spin-out, kind of classical, come out to Silicon Valley, sleep on a couch, raise capital, built that up to 50 people and exited the company to Roche in 2015. It was synthetic biology before that was a term anyone knew they wanted to use, and it was when this was a differently named conference, so it's kind of a cool background, so really happy to be here.
Wonderful. And maybe, Gary, if you can lay some context and foundation for our chat today and perhaps give us a definition for precision medicine and a little brief history of the space, if you will.
Sure. So I always look at precision medicine depending on what era you're in, right? So when I first starting out at Genentech, I'm dating myself, I went to school at UC Berkeley in the 90s. So precision at the time, say for instance, growth hormone, right? You could call it to be a precision medicine because based on the phenotype, right? You're short, you cannot grow, you got growth hormones, right? But that, to Chas' point, is kind of like a one size fit all, right? You're based on phenotypes. Over time, I'm just using Genentech as an example. You could see Genentech Roche as evolution, right, of the definition of precision medicines. And then later on in the late 90s, you could start to see HER2, right? The Herceptin is become more precise because that's overexpression of HER2. So that is one size, right, in that you could see. But even with that, then over time you see like people start to leverage the patient's own immune system, right, to fight against cancer. That's where you see the IO drugs start to come to the market, including PD-1, PD-L1, which have remarkable efficacy. You're the lucky 25%, it worked really well for you. But there's still 75%, right, that does not work. So the question always in the industry have spent the past 10 years trying to figure out which 25% of patient work, right? Also depend on indication. So that to me is start to hone into the precision medicines. And of course you always have, genetic driven like mutation based, right? Like the EGFR mutation type of patient that you could see and screen. For those, usually you could see 40 to 50% of patient will work, but still you have half the patient does not work, right, in here. So the question is like, what is really driving that? So that's where company like Roche, right, invest heavily buying company like Flatiron or Foundation Medicine, right, trying to dive into the patient data to use AI algorithm to see whether they could fish out those patients, right? So one approach to precision medicine is use big data, AI-driven. But another approach would be like what Xilis is trying to do in here is to take the patient-specific tissue, right, or tumor, and then you try to preserve that tumor microenvironment, and then try to test the drug ex vivo to see whether it works or not. To me, that is kind of like the ultimate precision medicine before you test it on humans, right? That's as close to individualized medicine as possible. So if you look at it, precision medicine over time is kind of like a funnel. It's getting smaller and smaller, right? Now at Xilis, we're trying to do it patient by patient basis to understand whether a drug works or not. Oncology is very important, right? Because most patients only live for a couple of months if you're in stage 3, stage 4. So knowing whether the drug works or not quickly, because usually it takes about a couple cycles before you know the drug works or not. By then you could be dead, right? So if we could use ex vivo approach, within like literally 2 weeks, we could give you a pretty good chance and confidence knowing whether the drug works or not. That to me is the ultimate definition of precision medicines. So that's how I see the evolution happen over time.
Thanks, Gary. And Jason, maybe if you can have you build on that and talk about kind of the modern archetype of precision medicine companies and some of the examples of those working in the space today.
Yeah, no, it's great. We actually, it's good to pivot off of a bit of a shared Roche background, and it's kind of what was the synthesis for my own company at Endpoint Health now. I've always stood on the outside looking at oncology, and you mentioned it perfectly. You've got Flatiron Health, so there's companies that tend to look at how do I dig into data to discover some new signal, something that gives me insight on where's the 25% I care about, and equally important, what do I do with the other 75%, 50%? What is a therapy or group of activities that could help those particular patients? You do have organizations like Foundation Medicine and many other that tend to translate those insights into something actionable, an actual test that can be ordered by a clinician quickly or longly, depending on the type of disease, to rule in or rule out a particular therapy. And of course, you've got the actual therapeutics involved. I think what's kind of interesting that you're seeing now is, I'll pivot us away from oncology. You're really starting to see precision medicine and in today's definition, extremely advancing the oncology space, but also spilling very aggressively outside of oncology. If anyone follows, the last 2, 3, 4 years, you have companies like Myocardia, which really was probably one of the larger acquisitions right into BMS, looking at defined patient populations for heart disease. You're moving into areas like neurology. You've got Neuroma, Denali, and a bunch of other folks, people looking at kidney disease. And where Endpoint Health sits is in immunology, which, funny enough, the only thing we don't touch in the immune system is the oncology space, but we're looking very broadly at how can we redefine what immune-driven illnesses look like. And I think what's very interesting about combining those 3 archetypes, data science, testing, and therapeutic development, is when you get into very complex diseases that have not had as much progress as oncology, we're kind of at those first steps of combining all that information to really define, do we see fundamental underlying diseases that are more real, that actually will drive treatment choice, that are distinct from whether you have swollen joints and knuckles, whether you have an infection or not. And really through combining all of those pieces, you can actually pick those patients out, make the test to find them, and then actually drive clinical trials to the right therapy. I think that's going to be a really interesting space, and we're all catching up with our far ahead brethren there, but I think it's a new area that's exciting.
Yeah, and we're big believers of this combinatorial approach at Alix Ventures. As Jason mentioned, the data-centric component of longitudinal patient data, combining that with diagnostics and the precision therapeutics, something that we feel is really a patient-first approach to precision medicine, and proud to support Xilis and Endpoint as they're at the forefront of this revolution here. And maybe, Gary, if you can share more about Xilis's platform and the ecosystem you're building and how that really works.
Sure, so Xilis have a platform they call the MOS, microorganospheres. So what they do is basically take any tissue, it could be tumor, cancer tumor tissue, or it could be lung tissue, a normal tissue, and then we find a way to grow them into a small droplet size of ball in here that we retain the tumor microenvironments or your immune system. So what that enable us to do, right, 2 applications we're exploring quickly right now. One is to use it for diagnostic purpose. You could imagine if you could depose those droplets right into microarrays, you could then test multiple drugs, different dose level in a very quick way to see whether it worked in a specific patient, because those droplets, right, those patient-derived droplets is patient-specific, so you could see whether your own body immune system fighting against that or not, or whether the drug will work on the patient or not. So another application we use is kind of moving it to earlier stage, right? Instead of just using diagnostic to guide the treatment, you could use the same platform to do drug discovery. So right now we are having significant conversation and partnership with major pharma company to do compound library screening on the compound on multiple, right, patients-derived tumors at the same time. You think of it back then, or not even back then, like mostly use a 2D cell culture, well known. Oftentimes you see what happened is sometimes those drugs work in certain cell lines, right? But because the patient type, the cell line is so much limited compared to what we could do in our mouse platform, that whatever that say work in those cell lines may miss the larger populations, right? So when you actually do clinical study, you see them fail, or vice versa. It could fail in that cell line, but actually may work in other patient types. So with our platform, you basically, instead of limited to, say, 10, 20 different cell lines, you could test hundreds of patient-derived specific tumors that you could test them out. So I think with our platform, it suddenly expanded the possibility of discover drug that used not work in certain cell line or fail in other cell lines that now we find to work. So this is pretty powerful tools that we have in here, but also this going to chess points about, right, we're generating patient-specific data. Those data then fit into our AI algorithm, right, to help pharma company to figure out new drug, and then this cycle just goes cycle back and forth to give you new data sets that you could use for both guided patient treatments and also to discover new drug in here.
And Jason, as you said, kind of this precision medicine revolution is not just exclusive to oncology, but really now being pioneered in immunology as well. Can you share about kind of the patient-first ecosystem approach you're taking at Endpoint Health?
Yeah. So it kind of starts pretty simple. I think everyone has seen a commercial for Humira. So Humira is the #1 selling drug on the planet. I think $20 to $30 billion every year. For the biggest illnesses that Humira is used on, it maybe has a reasonable amount of efficacy in 40% of the patients, maybe 50%. And these are debilitating illnesses. You're talking about patients unable to pick their kids up, not able to lead a normal life. So this is the kind of opportunity, I would say, that you see with the immune system. So the question is, well, how can you find the 50% or 25% of patients that those therapies work for, and how broadly could such a solution work for? So what we focus on at Endpoint Health is solving that problem. We capture mostly gene expression data from patients that suffer from immune-driven illnesses. And so this ranges from everything from sepsis to respiratory failure, which was both really big problems in COVID-19, into chronic inflammatory illnesses like rheumatoid arthritis, inflammatory bowel disease, even some neuroinflammation. And what we do is use that data and run it through our proprietary platform, which really is asking a question: are there fundamental subgroups of patients? Are there really distinct immune subgroups that are common amongst these diseases, that those are what you actually want to treat? That you're not really treating a rheumatoid arthritis patient, but you're treating a patient that in fact has some high level of inflammation driven by something specific. And can we use that to pick out the right patients, not just in one indication, but in many, many, many different immune-driven illnesses? And what does that do? It creates the ability to have trials that have double the chance of success on average. You can also look at shrinking trial time, cost, but most importantly, what you're really doing is opening up the ability to treat patients where there's no therapies, like in critical illness, because you can pick the patients out that you need. How much would we have loved to have a great anti-inflammatory for COVID for all those ICU patients? You're also picking out the right therapy for each patient when you have chronic inflammatory illness. So the way we end up translating it is, we capture these large sets of data, discover these fundamental subgroups. We have some great publications out, but practically speaking, we build blood tests that actually pick those patients out in the real world and use those to identify the patients we'll enroll in our clinical trials. Our particular model is a little bit different than the typical platform company. We actually in-license and develop therapies ourselves. We do actually work with some partnerships, but we're mostly really building a new standalone therapeutics company, but really with a full precision, it's 130% precision focus. Every single therapy is developed to be targeted to the right patient across immune-driven illness.
And Gary, maybe to expand a little bit more, you touched about flywheels briefly, and incorporating kind of this real patient-centric data into your platform, and that being a real guide for discovery, development, and targeted delivery of therapeutics. Can you maybe touch on how that flywheel works at Xilis?
Yeah, sure. So, like it's first of like, think of this concept about the flywheel, right? Actually exists for a while. At Genentech, they always talk about flywheel being from the lab to the patient's bedside, and then from the bedside, right, back to the lab. So this is kind of like model exists. The challenge before was like you could only really see data at best, right, genomic-related data in here. At Xilis, what we're doing in here is patient-specific. So we literally, in a short period of time, have massive amounts of data, right? Because we capture not only like NGS-related data that you could sequence, but actually we capture imaging. We could literally see how the drug is working, right, on the patient's immune cells, but also on the tumor cells in real time. So we literally have video, if, next time, maybe next conference, we could show you some of the cool video, right? What you could see then, different, you think of for NGS, you're just sequencing it, right? You only have one data set. It's also not time series. Versus for Xilis, we actually could see how the drug works on a patient on a time-course basis, which is tremendous, right? So our business model in here is we do diagnostic, which allow us to generate tremendous amounts of data, right, that fit into our drug discovery engine, and then we can also see how drug react to patient-specific tumor and immune cells, and then that fit right back into the diagnostic side for us. So over time, we start building out, right, the functional information, and now we just build the AI engines, similar to what Jason wanted to do in here, is how do you tie all these pieces together? I think like right now, if you're in AI or machine learning, this is like good time for you guys. You could command huge premium because every company at this stage, right, collect tremendous amount of data. We need to understand what that means such that we could feed back into the practical application in diagnostic and drug discovery. So that's how I could see the flywheel is working really well.
And Jason, can you talk about that flywheel again at Endpoint Health, and perhaps maybe share a little more about the novel clinical paths you're taking and how you're doing therapeutic development on that side of things?
Yeah, so for us, the flywheel, it's actually kind of practical. We made a very specific choice to own our diagnostic, to own the development of our diagnostic, and actually be able to capture data that comes out of it. So if you can imagine a three-step process, we capture patient data, work on, understand that data, find, let's say, that a disease we used to think was homogeneous is actually made of type A, B, and C patients. We in-license and develop therapies for, let's say, a type A patient. You take those through clinical trials. They end up theoretically getting approved. We all hope for that. They go into the real world, and the beauty of having a test that's actually your own and attached to the therapy that's also your own is you can create a feedback loop. Now, practically, you can do that either through patient registries. We can, we don't pull in the patient-specific biopsy material, but we can capture data from in the field. So that's actually how we do our work now, observational studies. And what it allows you to do is really course correct. Any machine learning platform will get better as you ask the question, did I really pick out the right responders? If I thought there was a type A patient, were the responders really in an identifiable type A1 patient, and actually type A2 patients, they should have some other therapy. Maybe there's something that's randomly used in the real world that might benefit them. This is actually how we've discovered some of our underlying indications. We've found places where generic drugs used on patients in hospitals shouldn't be used on patients that we do want to use a therapy for. I think, how does this translate broadly into, therapies that we develop? Sometimes we identify key targets or key therapies with our platform. We can see that with the way we profile immune patients. We also can identify where known targets that have therapies that are already on the market could be known targets where there's approved therapies, but for other indications could be expanded. That's actually how we identified our first therapy, and we're able to kind of skip the line ahead of all the safety work and go straight into a phase 2 trial. So we're a bit agnostic. The way we decide whether to find a preclinical asset or take something that's on the market to patients is where's there the right business, and how do I get the maximum amount of patient benefit as fast as possible into the market, they're all open doors for us to go after.
Thanks, Jason, for sharing. And Gary, maybe to bring us to a close here, any final thoughts you'd like to share? How can our audience get in touch with you and Xilis?
Sure. So in terms of my final thoughts, right? You guys look at the evolution of medicines. Now, like people talk about cell therapies, right? This is kind of like the ultimate precision medicine because you take your own cells, right? Identify the right one, you expand it, you put it back, right? So this to me is as precise as it could be. And then we talk about flywheel, right? For size, our technology fly into this new area as well in the sense that, you know, which cell you choose to expand, right? To a certain degree, you know, relying on knowing which cells are more potent and whether work on your tumor or not. So because the work we've done before now allow us to apply to this extremely precise treatments area, right? So our technology evolved along with the therapeutic size as well in here. So this is my closing thought. I think we're really getting to the point that precision medicine is so precise that it's patient-derived treatment and patient-divided guided treatments in here. You don't know how to reach me, go to xilis.net, the leadership team, you can see my name in there, or you could go to LinkedIn to find me.
And Jason as well, any closing thoughts you'd like to share? How can our audience get in touch with you at Endpoint?
Yeah, I mean, closing thoughts. I don't know, this is a really exciting time. I was just noticing the change in the title of the conference where, you know, it used to be SymBio Beta. I used to give this talk about 3D printing because in my first career in aerospace, we would do some 3D printing, and in, I don't know, early 2000s, it was $200,000 to print some part. Around 2010, 2011, it was $20,000. We paid some company up on the peninsula to make a little plastic component. 3-4 years later, I went, not knowing any form of CAD design, I went to the little 3D printing, you know, shop down the street, and for $200, I broke their printer, learned how to do 3D printing, and made something better. I mention this as a really interesting evolution because what that really captures is the industrialization of making hard physical goods. I think where we're really sitting at, and we're, you know, both Xilis and Endpoint Health and many other companies here are a good example of it, is the industrialization of biology. And I think we're an example. I know I am. I'm not neither a clinician nor a scientist by training, but still up here talking about building a fully integrated company similar to what Roche is at scale. Data, testing, therapeutics, all working together. And I think you're going to see a lot more of that as we kind of progress, and it'll be just really interesting how we switch from an industrial view where you try to figure out, can I do something, to what should I be doing if I can combine all these parts. So it's just a really fascinating time to be around. Getting in touch with me, really simple. Look me up on LinkedIn, send a note. I'm not sure if my email address is on our website or not, but LinkedIn's probably the simplest place. And thank you all.
Thanks again, Gary, and joining us today, and Bill Biology Conference for hosting us. Amazing to have leaders like yourself stewarding this revolution, precision medicine. Thanks again, guys.
Thanks, Alex.